everybody. Now we are going to continue the organization of eukaryotic genome itself and how it happens in the case of the chromosomes. That is, the features of the chromosomes are being explained. We had earlier explained that the DNA is being coiled and it forms chromatin fibers, and the chromatin fibers are further condensed together to form the chromosomes. Now, a eukaryotic chromosome is usually a molecule of DNA when it is associated with certain proteins. And all these, the DNA as well as the proteins are being together, and it mainly carries all the information and all the genetic information which is being needed for the replication or for the propagation of that particular species. Now coming to the chromosome structure, the first one shows a unduplicated chromosome which is being shown, and when the cell division proceeds, what happens, that single chromosome itself will duplicate and it will result in the formation of one more chromatid is being formed, and with these two are being joined together by a centromere. So sister chromatid is one of the two attached members of the duplicated eukaryotic chromosome, and centromere is a constricted region in the eukaryotic chromosome where the sister chromatid is being attached. Now, at the end of the chromosome, if you go to consider the structure of the chromosome, you can find here, okay, you find this is the centromere and then this is a chromatid, okay. At the chromatid, there are different regions called the origin of replication. See, there are different regions of the origin of replication. In the case of prokaryotes, usually you have only a single origin of replication. That is, this point from which the DNA replication happens. But in the case of eukaryotes, the number of origin of replications are being found to be more. And what happens is, at the end of the chromosome, we have certain regions called the telomeres. Okay, in the case of eukaryotes, you can find such structures called the telomeres. At both the ends of the chromosome, you have the telomere. Okay, and these are sequences which contain various repeats of the following sequence. Now, for example, at the 5' end, you have the TG repeat. Okay, X and Y can vary 1 to 4. Okay, it varies. Okay, and at the 3' end of the DNA, you will have the AC repeat. Okay, and this can be greater than 10,000 nucleotides in mammals, and the ends of the chromosomes are replicated by the enzyme called telomerases. And within this region, okay, after the telomere, in between the region of the telomeres are the what? Genes present. Okay, the genes are also being there, and you can note onto this figure, that is this type of dash, okay, line with two which is having like a bracketed line like, okay, these represents the genes. Now look throughout this length, okay, between the two telomeres, you can find that there are certain regions. Now this is a region of a gene, this is a region of a gene, this is another gene region, this is another gene region, so like that. And after that, see, look at this sign, the zigzag portion, it mainly refers to the repetitive sequence. So in between the gene sequences, there are also repetitive sequences. The genes are not being found to be continuous as we notice in the case of eukaryotes. That is, there are coding regions as well as non-coding regions. Okay, the genes could represent these repetitive sequence. It mainly represents the non-coding regions as well as the mainly called as a introns and all that. So they have to be removed during the process of transcription and all that. So in between, and here you can see that in between there are lots of genes, and at the end as well as in between the genes there are certain repetitive sequences, and even in the telomere region you can see that the repetitive sequence is being indicated. And at the center, the centromere is there where the chromatids are being linked together. Okay, and there are certain proteins called the kinetoproteins that will aid to bind, that will mainly help to bind to the spindle fibers during the cell division. We will be discussing that in the next sessions. Now, these telomeres have an association with aging. There appears that there is a relationship between the length of the telomere at the end of the chromosome and the age of an individual. The older you are, the shorter your telomeres are. And we have a certain length of telomeres that when we are born with, but as we age, the telomere gets shorter. So you need to think, does our lifespan is predetermined by the length of our telomeres? The studies are going on, you can explore it. Now coming to the region of the genes as well as the non-gene sequences. As I told you earlier, the genes are located between the centromere and the telomeres. And there are about hundreds to thousands of genes in the case of eukaryotes. And lower eukaryotes, such as yeast, the genes are relatively small and they have only very few introns. But in the case of higher eukaryotes, as well as like mammals, the genes have been found to be long and they have many introns. And apart from this, we have got various non-gene sequences which include the repetitive DNA. And the repetitive DNA can include telomere, centromere as well as the satellite DNA. Now let me ask you, we'll go into the gene structure detail. If you go to consider the eukaryotic gene structure, okay, there are certain regulatory elements at the beginning of a gene, and there could be a promoter or a enhancer. Then you have a exon 1. Okay, what does this exon 1, exon 2 and exon 3 represent? Exon 1, exon 2 and exon 3, these are the coding regions of that particular gene. Whereas the other ones like intron 1 and intron 2 are the non-coding regions. So, and only the region's exons will be transcribed and further translated into the what? The transcription of that would the mature RNA would only contain the regions of exon 1, exon 2 and exon 3. Transcription can happen of the intron 1 and intron 2 in the first primary transcript, but by further on, by the process of mRNA splicing, when the mature RNA is being formed, the intron 1 and intron 2 is being removed. And you can also see at the beginning of the gene, you have a start codon called the ATG, and you also have a what? Stop codon. And usually, every gene is being found in a eukaryote is being found to be monocistronic. That is, a gene is being a gene segment will code for one particular protein. But in the case of eukaryotes, what happens from one start itself, there will be any and there will be different start regions itself, different transcription styles, and they will be also called as polycistronic. And in the case of prokaryotic genes, you won't find these introns intruding in between. They just have exons just flowing like that. Introns are not being present in the case of prokaryotic genes. Then, coming to the chromosome number. Every eukaryotic cell has a specific number of chromosomes, and the sum of all the chromosomes in a cell is given by a type. Now, a human cell, we have about 23 pairs of chromosomes, and hence we call it as a diploid state. Now, there are different organisms have different chromosomes number. Now, fruit fly has 8 chromosomes number. Potato has 48 chromosomes. Cow is having 60 chromosomes. Visacha rat contains 102 chromosomes. Then, horsetail has 216 chromosomes. Like that, the chromosome number will vary for different organisms. This is the human chromosomes that we have. Of course, we have 23 pair of chromosomes. In a total, we have 46 chromosomes. And among them, you can see that X and Y, that is, these are the sex determining chromosomes. So the type of chromosomes, we have autosomal autosomes as well as the sex chromosomes. The paired chromosomes with the same length, shape and centromere location and genes, which do not cause or do not contribute to the sex of the particular individual are referred to as the autosomes. They are also known as the somatic chromosomes. Okay, they mainly are responsible for the body functions, as well as the sex chromosomes. They mainly are responsible for the what? the sex determination of the particular individual. In the case of a female, you have a X and a Y chromosome. Wrong. X and a double X is being present in the case of a female, and in a male you have a X and a Y chromosome. So these are the sex chromosomes as we told here. The diploid reproductive cell in a female which is having a double X, and a diploid reproductive male has a XY. So the sperms get only one chromosome, either X or either, in the case of females, either X. That's the only option. But in the case of male, it will be X or Y. So if the X and the X joins together, you get a, if the you get a female offspring. Here in this case you get female offspring. With the F and the if the X and the Y joins together, you get a male offspring. So thank you.